3, we are now going to talk about metabolic reactions, energy, reaction rates, and introduce ATP, adenosine triphosphate. So starting with metabolic reactions and energy. All right. Energy releasing and energy requiring reactions are what we're going to talk about, exergonic and endergonic. And we're going to talk about energy changes within these reactions and then something called activation energy. Basically, enzymes are biological catalysts that lower the activation energy, basically making it easier for a reaction to occur. So first we need to define forms of energy. There are two forms of energy that in physiology we talk about, and that is kinetic energy and potential energy. Mostly we're talking about potential energy, which then can be used for kinetic energy. And, example of a potential energy is ATP. It stores potential energy in the inorganic phosphate bond in adenosine triphosphate. And that high energy phosphate and ATP can be used to perform movement in cells or produce heat in cells or can actively transport something across a membrane. And kinetic energy also can increase with an increase in temperature. So as temperatures increase, kinetic energy also increases. Potential energy, it's stored so it can be later used as kinetic energy. And everything, the food you eat has potential energy. We call it calories. Our cells can't use that potential energy in glucose, sugar, lipids, triglycerides. It actually has to take the potential energy out of the sugar and transfer that potential energy to ATP, which it can then use for kinetic energy. So that is really the process of cellular respiration, and we will be talking about that next. So we adhere to the laws of physics, and the first law of thermodynamics is that energy in the universe, which is a closed system, is constant, meaning it'll go from potential energy to kinetic energy and then go back into storage, potential energy, and so forth. Humans, though, are an open system. That means we can lose energy in the form of heat. So we don't just recycle. We're not destroying that energy. It's being lost in the form of heat, but it's still a form of energy. It's not being destroyed. So energy can be created, but it cannot be destroyed. And that kinetic and potential energies are interchangeable in the overall energy system in the universe. Second law of thermodynamics is that energy always proceeds in the direction where it gets spread out. That is to say, larger molecules will break into smaller molecules. That's the direction of things, usually from a high to a low. We're spreading out the energy from a high to a low, like diffusion, from a high concentration towards a lower concentration, from a high to a low. Types of metabolic reaction. We talked about this earlier, anabolic reactions and catabolic reactions. Anabolic reactions are all reactions in which larger molecules are being built by combining smaller ones. So you're building things up, right? You lift weights, lift heavy weights, and you take anabolic steroids. And guess what? You're gonna build a lot of muscle structure, right? Because anabolic steroids, anabolic's all about building. An example, taking several amino acids, AAs for amino acids, and bonding them together to make a protein. On the opposite side, catabolic reactions are where you're taking a big molecule or molecules structures and breaking them down into simpler ones. An example of this is stored carbohydrates in, say, your liver. It's stored as glycogen. That is a bunch of glucose molecules binded together or bound together in this storage form called glycogen. If you need glucose to go into your blood, you have a catabolism of glycogen into individual glucose molecules. And then a second catabolic reaction can occur during glycolysis, in where one glucose molecule is broken down into two pyruvate molecules. Again, you're taking one larger molecule, 6 carbons, and breaking it down into 3 carbon molecules. And you're going to have 2 of those 3 carbon molecules. Again, that's catabolic. Molecules can possess potential energy. Take glucose, right? We eat sugar. Sugar gets broken down into fructose and glucose. There is potential energy. That's what we call calories. Calories are potential energy. So we count calories, we're counting potential energy. And yes, glucose has potential energy in its bonds, but our cells cannot use that potential energy. The potential energy in glucose has to be transferred into ATP bonds. It's still potential energy in ATP bonds, but that chemical process of transferring energy from glucose to ATP is called cellular respiration. So we will talk about that next. In this transfer of energy, reactants react to form products, and products can have energy being released along with those products. So you can have to add energy to the reactants to get them to be products. You can have reactants become products liberating energy. They're different types of energy reactions, and it's kind of denoted by this delta E for energy. And delta means change in. So it's basically delta E is saying change in energy. And so the delta E can be positive or it could be negative, meaning you had to add energy in or you released energy. And so exergonic reactions, those are catabolic reactions. It's a change in potential energy. It proceeds spontaneously, and the energy in the reactants is greater than the energy in the products. And so what happens is you release energy. So energy is being released. Endergonic reactions, those are anabolic, where you're actually building things up. And again, there's a change in potential energy, but it's not spontaneous. It needs an active driver. And so a way to say that is the energy in the reactants is less than the energy in the products, meaning you are adding energy, right? So you're inputting energy. Energy input is required to get to the products. And so the delta E is positive, meaning you have a positive change in energy. You're adding energy, right? So positive means you're adding energy, whereas negative means you are liberating energy. So exergonic and endargonic. And this matters when we talk about activation energy. So in reactions, there's something called an activation energy barrier. So reactants and products don't just form spontaneously very easily. And so that is the activation energy barrier, the barrier between those two reactants getting together to form the product. And so it is the energy barrier. And this limits how fast reactions can go. The higher the activation energy barrier, the slower the reaction. So we try to lower the activation energy barrier to get reactions to occur faster. And so the energy of the barrier is called the activation energy. It limits how fast a reaction can happen. And that is because you're waiting for those two reactants to get together to form a product. And so we get to this concept called rate. The rate is how fast reactants can become products, right? How fast can you consume or put those two reactants together to form a product? How fast are you generating a product? And reaction rates matter because our cells don't have all the time in the world. They need to do their metabolic functions quickly. If you are running a marathon or a race, you're taking glucose molecules, sugar molecules, and you're taking the energy out of that glucose molecule and you're transferring it to ATP molecules because your muscle cells can only use ATP to make them contract. And so if you're racing, there's a time issue here. You need to transfer that energy quickly. And so reaction rates matter, right? The speed of how fast those reactants can become products matter. And so proper physiology or bodily functions need reaction rates to proceed at a rate that matches the body's needs at the moment. If you're just sitting there on the couch watching Netflix, your reaction rates don't have to be too high. But if you start again running, the reaction rates need to speed up because your body needs more energy in the form of more ATP, and thus the reactions have to occur and put that energy in ATP quicker. So reactions, if rates slow down or are too fast than required, there can be cellular impairment. So if reaction rates get too slow, right? Hypothermia, you're super cold. Cold slows down molecules. Low temperatures, molecules move slower. You're decreasing your body temperature. Decreasing your body temperature is going to lower the energy of those molecules and thus the reaction rates. Cells won't get enough ATP generated, can lead to tissue failure, can lead to organ failure. So too hot denatures enzymes, slows down reaction rates. Too cold slows down reaction rates. And so we're going to go over three factors that affect reaction rate. So first is concentration of reactants. So when we have these brackets around something, it means concentration of. So concentration of reactants. How much reactant do you have? That's number one. Number two, how much product do you have? Right? Because this is concentration of product. You're not going to keep producing a product if you have too much of it. you have a high concentration in your cell of ATP, you're not going to keep generating ATP. There's already too much of it. It's as if you were a shoe manufacturer and nobody's buying shoes. You have a warehouse full of shoes. You have a high concentration of shoes. You don't keep producing shoes at that point. You try to sell shoes, try to get rid of that product. So that's number one. Temperature is number two. So as you increase the temperature, you're going to increase the reaction rate. If you decrease the temperature, you're going to decrease the reaction rate. Increasing temperature increases the average kinetic energy of molecules because it increases their activity or movement. And if they're moving more, there's a higher likelihood I should say probability of combining or colliding to form a product. So temperature does help. The other factor is the height of the activation energy barrier. So we did talk about this in a previous slide when we talked about this activation energy barrier. What is it going to take to get the reactants to get together? And so if it's a high activation energy barrier, then it's going to slow down the rate. If it's a low activation energy barrier, then it's going to speed up the rate. All right, unit of energy. Now remember, delta E is the difference in energy. And we use calories, joules in chemistry, but calories when we talk about humans in physiology. 1 calorie is equal to 4.18 joules, but we're only going to talk about calories. So 1 calorie is actually the amount of energy that is required to raise 1 milliliter by 1 degree centigrade. So that is a measurement of potential energy. 1 calorie is the amount of potential energy you have to increase 1 ml of water by 1 degree centigrade. I'm going over this because we talk about calories. So you should know what the definition actually of a calorie is. Rate of forward and reverse reactions are equal. No net change in concentration or products and reactants, and you have no difference in energy. That is what we call chemical equilibrium. So for it to be in equilibrium or be balanced, 1, there's no reaction happening. 2, there's no net change in the concentration of products or reactant. And 3, there's no change in energy. Well, the law of mass action is if you increase reactant concentration, it's going to push to produce more product. If you increase the amount of product being formed, it's going to push the reaction in reverse, right? Think about the shoe model. You have a high concentration of reactant. Those reactants here are actually becoming product as they react. At this point right here, it's equilibrium. You have the same amount of reactant as product. But if you keep combining reactants, you're going to keep forming product. Well, it's going to get into a state of equilibrium here. You're not going to form any more product because you already have enough product. But if you were to add additional reactant here, you add more reactant back up, it's going to produce more product until it hits a new equilibrium. So what am I trying to say here? The law of mass action is reactants are going to become products until you hit an equilibrium of having the same concentration of reactants and products. If those products are being used or consumed, then reactants will continue to become products because it'll never reach an equilibrium. Enzymes play a role in chemical reactions because enzymes are proteins. Chapter 2 is all about transcribing and translating proteins and then protein conformation. And we have lots of different proteins, but we're going to talk about proteins that are catalysts. And biological catalysts are enzymes. That's what we call enzymes. They're biological catalysts. And what do catalysts do? They increase the rate of chemical reactions. How? They decrease the activation energy barrier. How do they do that? How do they decrease the activation energy for a specific reaction? They actually bind to the substrates to bring them together to form product. I don't know. It's really kind of a dumb analogy, but enzymes are, I don't know, I guess we can say hinge or Tinder. I don't think anyone uses Tinder anymore. Oh, maybe they do. I don't know. Enzymes are like Hinge or Tinder. They're apps that allow you to see other people who are on that app that are in your location. So you can get together and meet for coffee or whatever. They're bringing two products together, or I'm sorry, they're bringing two reactants together. You're one of the reactants, the other person that you quote meet on Hinge or Tinder is the other reactant. And you can become a product, a couple. Would you have met that other person had there not been an app, i.e., an enzyme? Maybe, but maybe not. It could have taken years for you two to meet. Who knows? It's just the app, i.e., the enzyme, brought the two products, you and the other person, together quicker than if you just left it to chance. And so, really, that's what catalysts or enzymes do. Enzymes are specific to specific substrates or groups of substrates. They're not changed in the reaction. They're not consumed in the reaction. And by the way, enzymes always end in the suffix ase. So enzymes, anytime it ends in an ase. Lactate dehydrogenase. Anytime you hear ace at the end of it, it's an enzyme. But what I'm trying to say is Tinder is an app or Hinge as an app. The app doesn't change. It's not being consumed. It's not going away. It's not changing. It doesn't go anywhere. It's just allowing two reactants to get together. So enzymes are protein molecules. They're protein catalysts, but enzymes are biological protein catalysts. So enzymes a specific type of catalyst. Enzymes lower the activation energy and make biological reactions proceed at a higher rate. So the reaction rate is going to go up with the use of enzymes. To function, enzymes have to come in contact with reactants called substrates, and they form this enzyme substrate complex, right? So they bind to the substrate, become a complex, and again, they usually end in ASE. So enzymes are biological catalysts. They lower the activation energy of a reaction by becoming an enzyme substrate catalyst, right? Binding to it and bringing the substrates together to become the product quicker. Here is an example. You have carbon dioxide and you have water. And without the enzyme, you're just waiting for the CO2 to kind of bump into the H2O. But with an enzyme, it grabs the CO2 and the enzyme and the H2O. So it happens quicker with less energy, right? And it forms carbonic acid, which is the product. And so it has lowered the activation energy. The energy needed to produce the products has lowered, and the reaction happens faster. So enzymes lower the activation energy and to make reaction faster or product produced faster, however you want to say it. So key features of an enzyme. Enzymes bind with reactants, non-covalent, meaning they're not bound together. They have these weak interactions that keep them next to each other. It could be hydrogen bonding, could be ionic bonding, could be hydrophobic interactions. The point is, it's a weak interaction holding the enzyme to the substrates. Enzymes are also specific. So one enzyme is specific to a substrate or a group of enzymes to similar substrates. They are reversible. Enzymes bind, and sometimes the same enzyme that is going to take the product carbonic acid and break it down, so this would be catabolic, into carbon dioxide and water. That same enzyme can actually take carbon dioxide and water, anabolic, and produce carbonic acid. So some enzymes are reversible. Temperature and pH can alter the enzyme activity. Why? Because enzymes are proteins. And proteins function in a certain temperature range and a certain pH range. So that's number one. Number two is temperature can increase, or if it goes down, decrease molecular activity. And so the molecules are going to move at a different speed. So temperature and pH can alter these things. Cofactors and coenzymes. Ions and molecules required by enzymes to become active are called cofactors. So there are ways to activate an enzyme. Sometimes enzymes are not active until they have a cofactor. And so you can have trace minerals. Magnesium can be a cofactor, zinc, iron. These bind to the active site on the enzyme. To activate enzyme. Coenzymes, also vitamins, they can also activate enzymes. So coenzymes are organic molecules that transfer chemical groups during chemical reactions, and they could be derived from vitamins groups. We don't get into that too much, but if you go on in physiology and biochemistry, you'll learn more about that. Enzyme catalyzed reactions, factors that affect rates of enzyme catalyzed reactions. How many molecules are generated per unit time? That is the definition of rate. Right? So how many molecules are being generated per unit time? That's the enzyme's rate. And so some reactions generate thousands per second, others take minutes. So enzyme catalytic rate can be different amongst different enzymes. Another factor is substrate concentration. How much substrate do you have? We call this supply chain, right? If you have a high supply chain, you're going to have a lot of substrate. You can actually produce product. If the supply chain stops or goes down, supply chain issues, you don't have enough substrate, you're not going to be able to make the product. Second is, or third, I should say, enzyme concentration. How much enzyme do you have? Because enzymes bring substrates together. And if you don't have enough enzymes, you're not going to have enough gathering these substrates together. So that's going to decrease the rate. A fourth thing is affinity. Affinity is a measurement of attraction. How attracted are the substrates to the enzymes? If there is a high affinity, they're really attracted to one another and they are going to bind quickly. But if it's a low affinity, it does not bind quickly, right? Affinity matters. Then temperature, remember, temperature can increase or decrease, and pH can also alter the enzyme protein activity. So regulation of enzyme mediated reactions, these are these 5 things we just went through, and this is kind of showing you enzyme concentration, enzyme activity, how much substrate concentration, product concentration. These are all playing into the rate of how quickly substrate becomes product. So substrate concentration of enzymes in blood concentrations can change due to diet, absorption issues in the GI tract. Intercellular concentrations inside the cell can change as well. If the cell doesn't have the proper amino acids, transcription factors aren't being turned on to produce those particular enzymes. So substrate concentration of enzymes can change. When hopefully when you listen to Can a Workout Change Your DNA, working out or exercise, exercise can increase the concentration of enzymes in muscle cells, right? So you can increase concentration of enzymes. I'm kind of saying this a little too ahead of myself because this is enzyme concentration. You also have to have enough substrate, right? So you have to have enough glucose to then make ATP. It wouldn't matter if you had a whole bunch of enzymes if you don't have enough substrate. So let me back that up. You have to have enough substrate concentration, then you also have to have enough enzyme concentration. Oh, look, in this box, I have it here. Is that regular training increases intracellular metabolic enzymes, and so you can produce more ATP quicker. So that's enzyme production. If you increase the concentration of enzymes, you're going to use that substrate. So you also have to increase the concentration of substrate. If we're looking at these two, these reaction rates here. So remember, this is how fast reaction occurs. So it's real fast in the beginning here, but it's going to plateau. And so why does it, basically this is the fastest possible. That's that plateau. That's how fast the reaction rate can happen. Why? Because all of the enzymes are being used. So it doesn't matter, oops, it doesn't matter, this is increasing substrate concentration as you go this way. You can keep adding substrate. It won't matter if you don't have enough enzyme. And so if we look at this one here, what it's showing you is that this is the initial enzyme concentration X. So that's the first amount of enzyme. And all those enzymes are saturated here. But if you add more enzyme, you've added 2 times the amount of enzyme, right? Now it's going to proceed faster. Why? Because you have more enzyme to make more product. So you're going to be utilizing that substrate concentration. So the influence of substrate concentration on the rate of enzyme catalyzed reaction is, oops, let me move this. If we're looking at reaction rate, if you have a high concentration of enzyme, then your reaction rate is going to be faster, right? So this is faster, and you're having a higher concentration of substrate being used quicker. Why? Because you have more enzyme to bind to that substrate. If it's versus a lower enzyme concentration. So the reaction rate, what is the meaning of this? Basically, the meaning of this is the reaction rate increases with an increase in concentration of amount of enzyme present. So in that podcast, increasing transcription and translation of these enzymes in muscle cells increased the reaction rate of converting glucose or stored energy to ATP molecules to be used. Thus, you are increasing your metabolic rate. There's something called saturation, which is all of the enzymes are being bound to a substrate, and that's as fast as they can work, right? So if you have 100% saturation, it means all the enzymes have a substrate bound to it. And so when all the binding sites are occupied, it's said to be 100% saturated. If half the binding sites are occupied, it's 50% saturated and so forth. And so the concentration of enzymes matter, right? But also competition. And that's where affinity comes into play. The affinity is the how attracted the enzyme is to the substrate. So we get into the principle of chemical specificity. So chemical specificity is specific binding due to the molecule's chemical properties. So it's showing a ligand. A ligand is something that's bound to a protein. And it's a cation on this side, it's an anion on this side, and the shape of it fits. And so the cation is going to be attracted to the anion. The anion is going to be attracted to the cation. Cation is going to be attracted to the anion, and the shape fits. So this is going to have a high affinity. Why? Because it has the same shape and it has the same chemical properties. So if you're looking at this, this is a protein, and this is the conformation. And so you can see that these are the amino acids, but it's in its tertiary shape, which with all the folding, it ends up folded like this. Now, certain R groups are going to have a negative charge. Certain R groups are going to have a positive charge. And if the ligand has the right chemical specificity, it's going to bind. Affinity is how much specificity is there, right? So a high affinity is going to have all of the right shapes So shape is going to match and the chemical properties match. That would be a high affinity. A lower affinity would be you have some chemical and some shape match, but not perfect, right? So it's a lower affinity. So the affinity of the enzyme for the substrate is going to determine how quickly that enzyme binds to the substrate. So speed of binding of enzyme to substrate. And then temperature and pH as well. So uh, when you're looking at enzymes with a high affinity for substrate, you can say this is a low substrate concentration right here. So low substrate concentration. You don't have a tremendous amount of substrate, but it has such a high affinity for the enzyme that it binds most of the enzymes quickly. Why? Because there is a high affinity. If it's a lower affinity, to get that same amount of enzyme bound to substrate, it's going to take a whole lot longer and a whole lot more substrate concentration. This is one of the principles used to produce effective medications or drugs. If you can produce a drug that has a really high affinity for a binding site, you don't have to give someone a high concentration of that drug. You can give them a very low concentration of the drug, but it has a high affinity to bind. So you don't need to give them a lot of that medication. And so affinity is something that is kind of used a lot in biotech and biotechnology. So affinity is the relative strength of the ligand. Ligand is anything that binds to a protein. Um, and it is a measurement of how likely that ligand is going to bind to the protein. So high affinity, it's going to bind quickly and tightly. Low affinity, it's going to bind weakly and slowly. So this kind of shows you high affinity, got the chemical specificity and the shape. This has shape. This has neither. Just kind of smashed itself in there. So protein binding affinity, different proteins may be able to bind to the same ligand, but have different affinities. And so these different affinities are going to determine who gets to bind. If there are two ligands, this ligand is going to have the highest likelihood because it has a high affinity. And so affinity is of great importance when we're talking about physiology, especially medicine, biotechnology, because when a protein has a high affinity to bind to a ligand, very little of that ligand, i.e. drug, is required to bind to a receptor and cause whatever you want it to happen to happen. And so you can have less of that drug given to someone, and if you give them less of that medication, there's a likelihood that you'll have less side effects because when you flood someone with some chemical that is not normal in their body, it's going to bind to other things if it's in high concentrations. And so the influence of enzyme substrate affinity on the rate. Again, enzyme with a high affinity is going to mostly bind quickly, all of the enzymes, even though you have a low substrate concentration. There's also regulation. We talked about cofactors. There's something called allosteric regulation. There's also covalent regulation. And so let's talk about specificity. Here's the enzyme. Enzyme's going to bind to the substrate and form an enzyme substrate complex. It's going to make the substrate into a product. That enzyme then will release the product leaves, and you still have this enzyme to use because remember, enzymes are not consumed. And so this particular enzyme wants to bind substrate C. It will bind substrate P or substrate, sorry, that was product, substrate D, maybe B, but these are going to be low affinity. And this would be high affinity. So this is specificity. Oops. And so on the enzyme, you have something that's called the active site. The active site is where the where substrate binds. That's the active site. And again, it's important to understand the substrate's going to bind to the enzyme and become the enzyme substrate complex. Substrate can leave unchanged, but what's more likely is the substrate can be converted to the product leaves, and then the enzyme can be reused. So this goes through each step. By the way, a ligand is any molecule or ion that binds to a protein. And so we're going to use that term ligand a lot. Ligands bind with weak interaction. Again, hydrogen bonds, ionic bonds, they're not covalently bonded. So you get the protein bound to the ligand, becomes a protein ligand. And so it can be removed. Now, which ligand is going to have a higher affinity, ligand A or ligand B? Which has a higher affinity? Well, hopefully you said ligand A. Why ligand A? Because if you bring it in, the shape fits, the chemical specificity fits, and it's going to do a better job of the cations and anions attracting one another, plus it's got a better shape. So better, well, the shape's the same. Shape's the same, and better chemical specificity. So that would be said to have a higher affinity. And if we look on this graph over here, this is the number of bound ligands. More ligand A is going to be bound because it has a higher affinity or protein, right? So higher concentration of A is bound than B. Another concept besides affinity is how much of that ligand you have. So here it's going to be, I'm talking about affinity. Here I'm actually talking about concentration. So the more you have, the more likely they will all be bound to protein, right? So if you have a high concentration, they're going to be bound to the ligand. So in order for, two proteins to bind, they have to be close enough together and they have to have some affinity, right? So the chemical specificity or charges have to be similar and the shapes have to be complementary. So three factors that are affecting binding, again, review: chemical specificity, the affinity, and if there are available proteins to be bound to, right? So there still have to be proteins available Saturated would be there's no more. So cells can regulate their metabolic pathways by controlling how much enzyme is present, but also by producing modulators to those enzymes. So we're going to talk about those modulators. And one of the modulators is allosteric regulation. And so each enzyme that's allosterically regulated has two binding sites. One is the active site. This binds the substrate. And then two, the regulatory site, which is going to bind some kind of cofactor or regulator. And so it's going to be the modulator. So the regulatory site is where the modulator binds. A modulator molecule binds to the regulatory site, and it's going to either change the shape, thus the activity of the enzyme, or it can increase or decrease the activity. Generally, both alter the affinity or catalytic rate of that enzyme. So it's activating the enzyme. And so here's an example of allosteric regulation. You have the modulator, cofactor, coenzyme. It's going to then bind, the modulator binds to the regulatory site. And when it does that, it changes the shape of the enzyme active site to now be the correct shape for the ligand to bind. So a modulator molecule binds to the regulatory site, change the shape and activity of the enzyme. Binding of a protein to one of the sites changes the other, and thus can increase or decrease the activity of the enzyme. Sometimes these modulators can change the shape of the active site to disallow a ligand to bind. So generally altering the affinity or rate of that enzyme. Allosteric modulation makes it possible to have enzymes there in your cell, but not active, because you might not want them active all the time. So it regulates when the cell wants to activate the enzyme. So this makes it possible to increase the activity of a protein without having to increase the amount. You have that protein there, you can activate it. You can also have activators which will increase the reaction rate, or inhibitors which can decrease, right? So they work both ways. The second type, allosteric being the first type of regulation, covalent regulation is the second type, and it's basically either an active or inactive. And when you covalently regulate an enzyme, you are actually changing that enzyme. You are bonding something to it. Most common is adding a phosphate group. You bind that inorganic phosphate, covalently bind it to the protein or enzyme, and it activates. But you're forming a bond. And so covalent modulation creates a bond. And so when you phosphorylize an enzyme, you covalently modulate it. It's going to activate it. So this shows you, here's an enzyme. It's got a functional site. When you add an inorganic phosphate to it, here, it changes the active site to bind the ligand. Here, the functional or active site is the wrong shape. You covalently bond an inorganic phosphate off, off the ATP, it changes. You're using that energy in the inorganic phosphate to change the shape of the active site to become active. It's covalently bonded. And so covalent regulation is generally phosphorylizing and dephosphorylizing enzymes. So you're covalently bonding. Covalent regulation, kinases are the most common enzymes that add phosphates and covalently bond them, and phosphatase is the enzyme that generally removes phosphate groups from a protein. Feedback inhibition, conversion of B to C is the rate limiting state here. So if you have too much C, or the rate at which you're producing C, say you have too much C, it's going to come back and inhibit this enzyme. So this step stops. So feedback inhibition is when one product is the rate limiting step in the rest of the reaction chain. End product inhibition is if you have too much end product, it's going to come back and inhibit the first enzyme in this reaction pathway. So nothing happens, right? Again, it's if you have too much shoes produced and you have a warehouse full of shoes, you don't keep producing shoes, right? You stop producing shoes until you sell those shoes, get rid of those shoes. All right, onward.